Hello, welcome to my online lecture about scaling up bioimaging with microfluidic chips. I'm Tobias Wenzel, professor here at the Pontificia Universidad Católica de Chile, and in my lab we develop new methods based on microfluidics and bioimaging, and today I would like to tell you about some examples about how microfluidics can actually advance your bioimaging methods. First of all, what do we actually mean by microfluidic chips with which we would then work on the microscope? In this case, we mean these kind of devices that are usually custom made. There are other materials available, but usually research grade means these kind of PDMS chips with small channels inside where you can process cells and liquids and media. And these are usually made with photolithography and soft lithography. In some cases they can be 3D printed and other methods are available, but many times you need a lot of precision, and so you use photolithography, the same method you use for making electronic processors, for example. And so this is done by applying some kind of resin to a flat surface of a silicon wafer, and using a photomask and UV light to harden that resin, wash it out. And that way we have these kind of polymer channels here, yellow on this black wafer background. And these are basically 3D small plastic channels on a flat surface if you want. So then you can actually start making your replicas, your microfluidic chips in PDMS. So this is a two-component polymer that cross-links. So it's a silicon carbon polymer that cross-links into some kind of rubber that's similar to the silicone you use, whatever, to seal your shower. And so it hardens and you can detach it, and then you can reuse the master to make more of these chips. You do need another step though. You also need a plasma treater usually, so it's a little oven that fills with plasma light, ideally oxygen plasma, and that creates free radicals on the surface of the PDMS, and then when you attach it to a glass slide, it kind of covalently bonds and creates a really strong link to the glass. And that actually creates the channels that were previously on the surface of these PDMS materials, and then between the PDMS and the glass you get these channels. So this is the standard procedure to make these kind of chips like we've seen here in these examples. And with the photolithography you can have a very high resolution of 5 microns or less usually, and you can create very precise features that translate into channels. Okay, now that we know what these chips are, let's talk about the field in general. And so one thing to know about it is that it's an incredibly diverse field. It's also not very new, so it already has more than 40 years to develop. And in contrast to other fields that have more ups and downs, this is actually continuously increasing, and there are more than 6,000 microfluidic publications per year. So there's a lot out there, so impossible to read them all. And they span various fields of material science, chemistry, and biology, but particularly in biology, biological applications, there are also many, many things you can do with it. So it's really impossible to give a complete overview. So in this lecture, we're really just talking about a couple of examples that relate in particular to microscopy. And so I've tried to group some of these examples a little bit into 3 different levels of advancement. So you can actually already do a lot with very simple microfluidic flow chambers on a microscope. And yeah, currently in the project Libre Hub, we're running a workshop to for some people to test out microfluidics in different Latin American countries on their advanced microscopes. And there's a lot you can already do, but the more control you have over your experimental parameters by having flow control, gas control, different timing, advanced software, the more you can get out of your system and the more you can test. And so when you have different gradients and traps and gradient control, you have a more complex intermediate system that usually requires some additional equipment, like for example computer-controlled pumps. And then there's advanced use of microfluidics, which is something that our lab specializes on, mostly around droplets, microgels, organs on the chips, different interfaces, embedding materials, electrodes, membranes, and so on the chips. So that is a little bit more involved to create these chips and to operate them, but it's all possible. And also a lot of the advanced microfluidics, especially droplet microfluidics experiments, don't actually immediately relate to microscopy. So we'll keep that part, even though it's my specialty in a way, we'll keep that shorter in this talk because, there's a lot that can be done just to give you an idea, but a lot of it also relates to whatever sequencing or other kind of high throughput readouts that are different from microscopy. But there is a really nice overlap, and if you're already a microscopist, then this lecture hopefully gives you some ideas about how to scale up and improve your microscopy tools with microfluidics. So to jump in, we'll talk about flow chambers. So at the end of the day, it's really just, it can be a very simple chamber that can be a microfluidic chip, or even maybe 2 glass slides with a little bit of sticky tape around the edges as a flow chamber. So they can be quite simple, but yeah, using the PDMS makes it easier also to connect tubing and other practical aspects. And for first tests of using flow chambers in your microscope, you could even use something like gravity flow. So for example, having your liquid medium or so that you want to flow through the chip, just putting it higher up and letting it flow through the tubing, even if you may not have access to advanced pumps. But we'll get to some research, to some open source instrumentation resources that we developed at the end as well, in case you want to, for example, build your own high-end but low-cost equipment to do that. Okay, so flow chambers are great to observe all kinds of organisms, biofilms, plant roots, and phase transitions over time. And it's really good to do live cell imaging. So monitor cells over time, just gives you a little bit of control. You don't have an open system where your liquid evaporates or something, but you have a nice little cell that you can, like a chamber that you can monitor on your microscope. So in a simple case, you'd have a simple chamber and some kind of flow by gravity or pump, and then just a waste outlet on the other side. And you use whatever microscopy method you're already familiar with, and then perform advanced data analysis. To get most out of it, you usually do need a good microscope for this, for example, with a powerful autofocus, resolution, confocal, whatnot, and of course to make most of this data advanced image analysis pipelines. So this is really for people that have good microscopes and good microscopy skills, bioimage analysis skills, and get more control over their sample. So there are still a lot of issues you can run into. So in this workshop that we're currently running, we're going to help different scientists to troubleshoot some of these things. But there's actually a very nice publication as well from a colleague in Germany who tried to outline and go into details of different issues that may arise in different imaging examples with flow chambers when you cultivate your cells on the microscope. There's some tips on how to get around it. So this is definitely a resource I recommend to you if you want to try this out for your research. Right now, we're jumping into a couple of examples. So here we have a microfluidic chip that's pretty simple. So it's just a very simple microfluidic channel, and it's mounted on a confocal microscope. So you get a 3D view of the channel. And in this case, they let a biofilm form on the surface. In this case, they also coat the glass with PDMS so that, that's not true, actually, sorry. In some other papers, and that is being done, so that the material on all sides of the chambers is the same. In this case, there's a glass bottom and the PDMS all around, which is sort of the standard configuration of our chips. Okay, but in this particular paper that is here shown as an example, what they do is that they actually have a relatively complex flow control. And so they change the frequency of different nutrient inputs to the biofilm and see how the biofilm develops, and also at which flows it's sort of washed out and when the adhesion stops. So this is something, there are a number of different papers, even without this sort of frequency oscillation, that also look at the biofilm adhesion strength, for example, by using the flow as a mechanical cue. So this is definitely something that can be done with different kind of cell types. And in a particular case of looking at bacteria, so this is not for biofilms really, but more for monitoring the physiology of bacteria and their genetic expression. There's a very popular chip design that's called the mother machine. In my view, it's still a flow cell. You could also view these things a little bit like traps, but the idea is you have a main channel, and then of the main channel, we have really, really small channels that are sort of 1 or 2 micron broad usually. So this is actually very hard to fabricate, and sometimes you can't even do it with photolithography. Often there's used e-beam lithography, which is more expensive to make these kind of very high precision channels sometimes. But the key is that the organism that you study, in this case Escherichia coli bacteria, that they just about fit into these little side wells. And so then by chance or by centrifugation, some cells enter, and when they grow, they sort of push towards the end. And then once a cell reaches the end, that cell, even when it divides, there's always a cell that stays there. So it's called like the mother cell, and you could look at the physiology and genetic expression of their offspring. So you can look at their, for example, the genetic sort of hierarchy. So the mother cell always stays at the end of the channel, and you can look at, in which what do the cells look like that they divide, the division rate, the cell size, and so on. And in the case here above in this video, they implemented a genetic oscillating circuit that expresses a fluorescent protein, and you can just characterize this circuit on a single cell level with this kind of flow chip. So the mother machine is a famous, well-known chip for that, and actually you can extend that also to other organisms. So there are designs available for using, for example, Bacillus subtilis, or yeast cells on these chips, and they require slightly different designs and sizes of these channels for it to work. But there's literature out there to get design ideas. Okay, next, one very nice thing you can do when you already have your microfluidic channel available is to actually work with gradients. So microfluidics, these microfluidic channels, they're small, which means that there's a low sort of Reynolds number on the liquids, on watery liquids especially, which means that there's a laminar flow and things don't really mix very well. But you can create these kind of mixing trees to create gradients. And in this paper, for example, they do this to create a number of imaging chambers in parallel that can be loaded with eukaryotic cells. So in this case, it's sort of nerve cells, and you can image how they react to different concentrations of certain drugs or chemical stimuli, for example, and monitor them over time. So here you have an array of growth chambers with a whole sort of dimension of a condition. So concentration gradients are nice. You can of course also use other gradients like temperature and so on. We'll get to some other examples. Or you can directly make a gradient in the chamber. So instead of having separate chambers with the discrete mixing values of the gradient, you can directly inject the cells into the middle of a channel that has a gradient, for example, to study the chemotaxis of motile cells, or in general sort of how they react to certain conditions. But this is a constant flow, so you would have to constantly flow in new cells and see how they react in this particular chamber design. What else you can do with this laminar flow, so instead of creating a gradient, you can actually use the fact that they mix very slowly. Of course, it depends therefore on your flow rate, how fast they mix. So if your flow is very slow, you will get more mixing, and if it's relatively fast, as you can see here on this cover image on the right, you get these very discrete lines of different aqueous solutions that don't mix, because they'll mix further down the channel, and they mix by diffusion only. Yeah, so it's not a mixing by tubular flow and sort of vortex generation, but it's really a mixing through diffusion of the molecules, and so it depends on the molecule size how well they mix. So in this case, one application that you can do is you can have two separate channels that actually combine in some area, but they just flow parallel, and the smaller the molecules, the further they will diffuse into this other buffer, and you can use this to refresh some kind of cell medium, for example, in your culture chamber. Just to mention a couple of ideas of how you could use this in your microscopy system. Right. Then you can control any factor that you can think of really in your microscopy chamber with microfluidics. So in this case, it might be difficult to have a very precise gas control also in space in your system in a classical imaging chamber. So what these authors did, so this is a lot of information here on the screen, but you can look at the paper on your own. What I want to highlight is that they they connected different gas supplies to their microfluidic chip, and they basically created an additional channel around their imaging chamber on the microfluidic chip. And so the gas flows around it, and because the PDMS actually allows a gas to go through, it's gas permeable to some degree, since these channels are close by and the distances are small, you actually can create a pretty good gas exchange between your media and the channel where you flow in a gas. So they have a very good control over time and space of their gas that they apply to the chip. And so they analyze the growth of their cells over time under different gas conditions. And these images here, I think, are really, really nice example of how these chambers, I don't want to go ahead yet, of the data you get and how this is analyzed. So especially when you don't use this mother machine design where you can quite easily focus on a certain channel and the offspring cells, you get this chamber high sort of 2D arrangement of the cells and their genetic expression. And then it's really up to using your data analysis pipeline to get good information out of this. So you need to have segmenting and quantification data pipelines to make most of it, and also to get these high resolution images, you will have to have a nice microscopy setup and a good autofocus. So those are important things to consider, and this is the kind of data that you usually obtain. So as these are bacterial cells, they're very small, and this is a high resolution. So depending on your cells that you study, this may or may not be a bigger constraint. So, but this means that beyond flow and gradients, you can also control gas and many other things. In this paper, they give a couple of examples of different manipulation methods of different model organisms that you could see. So you can create, a sort of artificial porous medium model, for example, and look at how your model organism or worm, for example, behaves over time, moves through this kind of terrain. You can do chemical stimulation over time, injecting different stimuli to a trapped larvae, mechanical stimulation including, so you can have chambers also with gas pressure basically that increase or decrease in size and therefore stimulate the trap of the organism. Or also, instead of using chemical gradients, as I mentioned before, you can also, for example, control the temperature of the liquids that you inject, and you can create temperature gradients if that's relevant for you. Or again, do genetic perturbation and sort of single cell transcriptomic imaging over time, for example, on your traps. So there are really many things you can do in different ranges of complexity and depending on the equipment that you have available. You can also create more complex multi-layer systems that are really actuated and so on, but the core feature here is for data generation. You really want to be imaging the system over time. So this is, again, we're focusing on the interface between microscopy and microfluidics in this little lecture. So this is a very nice chip where they mentioned to look at the microbiome host interactions, but not doing this in a mouse, but doing this with human cells on a chip model. And and here they also control the oxygenation, so they have some kind of oxygenized medium flow here that is equivalent to the blood flow, and the whole chip gets placed into an sort of anaerobic or microaerobic chamber incubator, You have different levels of this chip and a trap membrane where the cells are placed, and you have chambers on the side that can contract and create some kind of movement of your cells, which is important for the gut cells to form correctly and for these epithelials to really create a membrane that is physiologically representative of the gut. So yeah, this is a very nice, complex, advanced system that you can still image through and create relevant data. Also, this is a nice example that I like, that here you have a number of imaging chambers in parallel that are actually connected. And the idea is that here in different chambers you can cultivate different organoids representing different organs of the body. And because the conditioned media, the supernatant basically from each of these can reach neighboring imaging chambers, there is a metabolic interaction between these different organs, and you can study more complex human on a chip models in this way with based on metabolic interactions. So there are really a lot of things you can control in terms of times and space using microfluidics on your microscope, and you can parallelize things in a much more custom way and with more throughput also than normal multi-well plates, for example. Talking about multi-well plates and the difference to microfluidics, so you could see in some cases multi-well plates of some kind of traps, but you have a limited number of wells there. But you can also actually trap different cells. We were talking about these gradient flows, for example, so you can either continuously inject cells there to see how they react, or you can actually trap cells and then create a gradient on top of them, for example. So we'll just look at a few examples now of trap chips. So one that we are working on now is nice trap examples that are reversible. So here you can flow little spheroids or organoids, for example, onto this little trap chip, and whenever a spheroid of the right size hits one of these little channels in between these barriers, then they get trapped and there's no longer a flow through that gap, which means that no further spheroid is trapped there. And so you can trap spheroids in many of these traps, but some might remain empty. I mean, it depends on how big your sample is and so on. But the key thing is that as long as you maintain a slow flow from the opposite direction of the traps, these spheroids remain trapped and you can image them. But you can also wash them back out. So when you create a flow from the other direction, these traps basically no longer hold these spheroids, and you can recover the whole sample outside of the chip, and you can inject a new sample or the same ones again. So these are sort of, well, you don't normally reuse these chips as there may be some kind of biological leftover and cross-contamination. So it's not really for reusing, but it's for sample recovery of these important patient samples, for example, where you work with small material. You can, of course, also create microfluidic traps where these get permanent stuck, and then you don't need to maintain a flow, and maybe you have less risk of experimental error that something moves or gets out, but you cannot recover the sample. So this is an important example for sample recovery. And these sort of hydrodynamic traps are very common. And so here is another one for zebrafish embryos. And what's also special about this particular paper is that they created a second layer on the chip. So you have one layer of channels, and then on top of it you basically bond another microfluidic chip if you want that has a large chamber. And in this case they apply some kind of vacuum to it. And because, as we mentioned before, PDMS is partially permeable to gas, this aspiration of gas from the second layer means that any kind of bubbles that might enter through with the medium will get sort of sucked through the walls of the PDMS on the top, and you avoid those bubbles to interfere with your experiment. So this is one way of implementing a bubble trap, but there are also many others. It does require, of course, additional instrumentation. Here's some kind of vacuum and a more complex microfluidic chip, but these things are possible. Next, we have an example of, again, zebrafish traps, but not the embryos, but real small zebrafish. So these are much bigger traps, and this can be 3D printed. And so here, the authors designed this chip where you create an inlet flow, and you have a whole row of these different traps where you can trap little fish inside, and then you can expose them to antibody staining cycles, for example, or other media conditioning, and observe them on your microscope over time while you have these dynamic conditions. And without washing out the embryo, obviously, and not embryo, the little fish. And you can see its behavior and biological reaction. After zebrafish, our next examples are plants. So you can actually also grow plants on microfluidic chips. These are not that micro anymore, so these are relatively big structures, but you still create a nice tight imaging chamber, and you can actually grow the roots of these Arabidopsis plants into the chamber. For example, here they have a little agarose plug in a in a pipette tip, and they seed the plants there, and they grow into the channel, so that you can get very nice images. And you can, of course, control the chemical environment by adding different media conditions and add bacteria, for example, as in this case, to look at the root colonization through fluorescent markers. So this is also a very nice platform to parallelize and control your experiments and really get nice bioimaging data out of it. So with that, we conclude our examples of different traps of different organisms. Again, this is not all there is. These are just really some pretty random examples of things you can do by trapping organisms and controlling conditions and parallelizing your microscopy experiments. So with that, we come to the last part of the microfluidics content, which are droplet-based high throughput assays. This is something that my laboratory specializes on, but many of them are actually not imaging based. So there are other high throughput ways to generate data, which is more flow cytometry, analogous, or through sequencing, for example. And here we focus mostly on examples that are related to using microscopy on droplets. But there are a lot of interesting advanced assays that you should know about that can also be performed with microfluidics. And so what means droplet microfluidics? It means that in these microchannels that you create, these are normally pretty small in nature, you don't just inject your cell suspension, for example, but also oil. These are specialized oils that have been developed with surfactants that generate these very stable droplets. And because of the fluid dynamic conditions in these microchannels, this is a very controlled generation of droplets, and you can generate droplets very fast, and they're all very similar to each other. So you create these monodisperse emulsions, what this is called, and this is very fast. So here, this is a video with taken with a high-speed camera. So really, we generate 8,000 droplets per second. So in less than an hour, you can create millions and millions of droplets, and every droplet would basically be an experiment. Now, by dilution of the cell suspension, for example, you can create single cell encapsulation. So this is often used for single cell analysis, but you can also create molecular assays, interactions, and many kind of other controlled experiments. And one droplet would basically be one experiment in this case. And this is really the gold standard of high throughput experiments, much, much more scalable than pipetting robot experiments. A typical experiment looks like this. For example, you still use a microscopy stage, but even when you don't always really do bioimaging. Let's say you don't always perform data analysis or capture images, but in this case, this would just be to monitor the droplet generation, that everything is working fine. You inject oil. This would be the microfluidic channels here on the right. So you have different inlets. These little symbols around the inlet just makes it easier to punch the channel and find the inlet place more easily on the chip. So this is really just a visual marker, and then you have a little filter so that when any kind of little dust particle also comes in, it doesn't clog these very well-defined channels. And so you have an oil inlet, and then you have your cell suspension and sometimes something else. Like in this case, for single cell transcriptomics, you also have a bead suspension. So the cells and the beads get mixed first, and then they get encapsulated into droplets by meeting with the oil and collected. And now you generate single cell encapsulation here by dilution. So this means that a lot of droplets actually remain empty, and some droplets will only have a cell, and some droplets only will have a bead. But the dilution is chosen such that the probability of having more than one cell and one bead in a droplet is very unlikely, and only those droplets that actually have both a cell and a bead generate data. So you can still generate hundreds of thousands of data points in one experiment with this high throughput method. And the advantages of performing these droplet microfluidic assays are not only this ultra high throughput, but also single cell control. We mentioned it already. Low contamination. This is very important for single cell experiments. These are often equivalent to something like a single molecule resolution experiment, so contamination can be a big problem if not performed in tiny droplets. It's very versatile because you can perform all these different operations with droplets. So you can generate droplets but mix things just beforehand. For example, for single cell transcriptomics, you can mix the cells with some kind of lysis buffer just before encapsulation. You can incubate the droplets on chip or in a line, or off chip. You can re-inject them, you can merge them later, add reagents, or sort them. So there are really many things you can do, but the more of these things you do, the more complex your experiment and the equipment that you require. So one of the things that droplets is nice for is in this paper is actually cultivation of bacteria here from the gut microbiome, not just bacteria, but also archaea, fungi, and all kinds of organisms that are part of the gut microbiome. And they found that if they actually separate the cells individually into droplets and cultivate them there, they can grow a larger diversity of the gut microbiome. And one of the reasons for that is that the growth rate of the different cells is very different. So in a droplet, you separate the fast growers from the slow growers, and also you suppress any kind of negative interactions and suppression of growth between the bacteria, so that you can give the slow growers and the sensitive cells time to develop. You can of course also do in other papers, you can study the interactions between cells and also create a larger diversity of growth by actually co-encapsulating cells that need each other to grow. There are also molecular assays that you can do in droplets. So this is droplet PCR, droplet digital PCR, or just digital PCR it's called sometimes, so ddPCR usually, the abbreviation. And this is a competition to qPCR, or real-time PCR as it's called, because usually you have a higher sensitivity and specificity even than in qPCR, and you get absolute results of your molecule numbers out of there. So this is not a single cell assay, but it's really a molecular assay. So you encapsulate the PCR reagent, your master mix, and your sample DNA into droplets. And because the reagents that are now available, these fluorinated oils and surfactants, they're really very, very stable. So you can literally basically boil these droplets in a thermocycler and they will remain stable. So you can perform the thermocycling in the droplets and image or use some kind of flow cytometer basically to analyze the fluorescence of each droplet. And so every droplet that turns fluorescent basically has one molecule of the target in there. So you get an absolute number of your molecules in a very sensitive assay. So droplet PCR is a nice method to have in mind for droplets. And here we have an example of these cellular interactions. So here they genetically engineered two bacteria to be dependent on each other because they are lacking the biosynthesis pathways of some amino acids. And they basically found that if you work with these small droplets, they're usually used for bacteria, 55 micrometer, for example. Often it's in the range between 20 micron and 100 micron for bacteria. You get these very strong interactions, metabolic interactions between these cells, and all the cells can grow well based on this interaction. But even already going to higher droplet sizes, like 150 micron, you get significant suppression. So this means you encapsulate these cells, but there are just a few cells in the beginning. This is a very important point, because if you actually work with patient samples, environmental samples, and you want to capture the diversity of this sample, it means that you have to dilute the samples and really go for single cell experiments or very few cells. And in this case, when there are few cells, metabolites get very diluted in the space. And already in these bigger droplets, you see significant drops in cells that manage to establish this vital interaction before they die. So this is a great benchmark experiment, but it really shows that droplets are a great platform to look at metabolic interactions or other kind of interactions also. And it can be used to co-culture bacteria that normally don't grow individual in the lab because they require a co-culture. And so the bottom line is that interactions are usually between cells are very diverse and complicated to study, and droplets are really perfect for that. So there is another really great high throughput single cell analysis method, which is flow cytometry. Well, and in fact, microscopy, you can also say. But the problem is, in an aqueous environment, metabolic exchanges in particular get very, very quickly diluted. And in a droplet, you keep them together in a small volume, and you can analyze them with microscopy or a kind of specialized flow cytometry. So we wrote this review where you can see a lot of recent methods that have been developed to screen, surface proteins, cellular interactions, the use of biosensors or growth enhancers through co-culture, antibody screening, and molecular assays that you can do through droplets. So there's quite a lot that can be done, and some of these things can be done with the microscopy analysis. All right, here's a very nice example relevant to our work that we currently pursue, which is a literature example of co-trapping two droplets that you can merge. And so you have your cell sample and you have a stimulus droplet, and these traps are designed on a multi-layer system and in a geometry where basically these two different droplets of different sizes, they get trapped at specific points of this trap. So you will always have a combination of a big and a small droplet. So now you can do this sort of one-on-one combination assay where you have a certain stimulus that you release on the cell. In this case, it could also be pathogen released onto a spheroid sample from patients. And then you can use different fluorescent markers on your microscope as a readout in this parallelized fashion to monitor the experiment. So it's a really nice high throughput interaction experiment in a microfluidic microscopy chip. Okay, there are also more advanced materials you can use. So instead of just generating normal, let's say aqueous phase water droplets in oil, you can actually use gel. You can gelify the droplets, or even create chemistry that creates a gel skin around a liquid core, also called semi-permeable capsules. So here on the right we have an example of semi-permeable capsules that we made, where we performed a whole genome amplification inside. So all of the glowing capsules are small capsules where a cell was present, and so the fluorescence you see is just the DNA molecules that are present, and this is like an amplification of this one molecule of genome that was captured. And these capsules are nice because you can then you can use these powerful molecular biology workflows in parallel. So you generate the high throughput of your sample by encapsulating the cells individually, but then you just have one Eppendorf tube with all your capsules inside, or in fact microgels, like on the right where we encapsulated a few cells and they grew into these microcolonies and in gel microdroplets from agarose, or here as a co-culture at the bottom. So with microgels or capsules, you can then remove the oil again and they maintain their integrity, and you can do things like lysis, staining, or genome amplification in applying them in parallel to all your separated samples, and they remain separated, so you can analyze them separately by microscopy or genetic methods, for example. Here's a very nice example where they've even taken it a step further. So they encapsulated different cell types into microgels, and then they did some kind of 3D printing where they joined the microgels into little fibers with which you can print 3D structures, and you can use sort of different microgel emulsions in these different fibers and actually make a 3D assembly of different cell types that you controlledly apply to your 3D structure, and then you can co-cultivate them. So this is still experimental, but it could be very cool in the future. Now, beyond using these different cells, you can also assemble cell-like structures. So there are very nice protocols to, instead of just generating water droplets in oil, creating thin layers of oil to create liposomes, giant vesicles, and other similar structures. And there are even some advanced microfluidic protocols where you use pico-injection in this case, for example, to add certain membrane proteins onto these liposomes to create cell-like structures for synthetic biology or other kind of screening examples. So there's really a lot that you can do, but again, many of the analysis that is performed here is not necessarily imaging, but can be. This is the video of creating these liposomes. So it's quite nice because in this protocol, you apply more oil than just to form like a membrane, but it splits off automatically so that you really create only this liposome, which is a nice little method. Okay, so this is really it. We just want to get an idea, an overview of different things that you can do with microfluidics to enhance your microscopy. And we want to close with a couple of open source tools that we developed in our lab that may be of interest for you if you want to start with some simple microfluidics to control your microscopy experiments better, but later scale them up to more complex assays and droplet microfluidics, for example. You might require some more instrumentation like syringe pumps. These syringe pumps, which is basically just a motor pushing the end of the syringe to extrude the water onto the microfluidic chip. With this you have a lot more flow control than a gravity flow, and you can also control different media at the same time. And these syringe pumps can be really, really expensive traditionally. They're also very bulky and difficult to apply to your microscope. But we picked up some really nice open source designs from the internet and improved them with a gearbox and a couple of modifications and a new controller design, so that you can really have very precise flow control, because normally you want to have a very low but steady flow applied to your microfluidic chips. So you can do that with this design, and you have this controller box for manual intervention. You can also connect remotely to it for advanced flow control, like for example flow gradients and so on, changing the flow rate over time with Python scripts. So this is so with this you can really build these low-cost syringe pumps that have very, very similar performance to commercial pumps that can be quite costly. So this can be a nice way to enhance your microfluidic workflows. If you want to go into droplets, you might be interested in our strobe microscopy platform. So we often use this in workshops and trainings and outreach to other labs. We often combine this with our 3D printed syringe pumps. This is just a small microscopy platform based on a Raspberry Pi and the Raspberry Pi camera, but that is synchronized with with a high power LED strobe to really capture very quickly images of these fast moving objects like the droplets that you would not normally see with a normal camera imaging. You would just see a line because the droplet moves much, much faster than the imaging rate. But by creating small strobes of light, you capture these very nice images. And so you can see and troubleshoot your experiment, and you can also use it for imaging experiments in progress, like flowing cells, so that you might miss otherwise. And yeah, this has been designed to be easy to fabricate anywhere in the world with 3D printing and standard components, and it's really ideal for microfluidic experiments because it's small and compact and with a clean surface. So it's just kind of what you need instead of big professional microscopes when it goes about sample handling of all these different fluidics to make the droplets. And then you can inject these droplets into an imaging chamber and place them on your microscope for confocal imaging or whatever later. So this stage you can build it with this really detailed step-by-step guide that we made, how to get all the components and assemble them. But reach out to us if you have any questions. We also have this advanced flow platform that we developed also to generate these microgels, for example. There are very few commercial instruments that are able to handle several channels and the high pressures that you might need to insert these different gels. So we have a recent publication where we use this to analyze the stability of plasmids in cell cultures based on the high throughput staining and imaging that these microdroplets allow you to access. And one other thing that's one of the most high-end instruments of droplet microfluidics that we work on is actually an open source droplet cytometer that can also sort individual droplets. So this is for high throughput screening and directed evolution, where we have set up with different lasers and single photon detectors, where we can flow droplets through a channel and analyze their fluorescence, and use FPGA computing to quickly trigger electrodes to actually select certain droplets for further analysis. You can find all these resources that we work on, our different codes and hardware instructions on GitHub of our lab and the Libre Hub project. We also share a lot of different 3D printable designs that we created for different lab instrumentations and tube holders, pipette holders, optics components, and whatnot. So feel free to check out our resources and download them to improve your experiments. And last but not least, we create this YouTube channel where this lecture will be hosted, in fact, and very nice tutorials for you to explore open source hardware instrumentation and imaging workflows. There's a very nice workshop of Napari for bioimage analysis that might be complementary to this lecture for you to get more out of your imaging data. So with that, thank you for listening, and we'll be in touch if you have any questions. Thank you very much also from our Libre Hub project with my co-PIs, and until next time.